Finally, let us talk a bit more about post-alignment processing. More specifically, let us talk about the filtering step, since the other three steps are relatively simple and have been adequately covered with just the first level of detail. There are many criteria which can be used to filter alignment results. These include screening out alignments against artifact sequences, alignments against noise, ambiguous alignments, and weak alignments. Many of these results can be omitted from the raw alignment results by customizing the reference genome and by configuring various software options during the alignment process. However, rather than omitting these results from the raw alignment results, it is usually more informative to filter them out afterwards, because this allows users to calculate the percentages of the causes of disqualification. For example, in a gene enrichment pull-down experiment, including the mitochondrial chromosome in the alignment process allows users to differentiate between truly unalignable reads and reads which align to the mitochondria. Results which are disqualified from the main analysis pipeline may also still be useful in side analyses, or may be salvageable under certain circumstances. Since the alignment step is usually one of the bigger bottlenecks in the pipeline, it is more expedient to include such results in the raw alignment data and to remove them later. Also, while I use the word removal to describe the filtration process, what usually actually happens is that a copy of the file being filtered is created. In the copy, the disqualified alignments are not included. However, the original unfiltered results are still present in the original file, which has not been altered. Therefore, it is relatively easy to backtrack if you regret removing certain results, and you don't need to perform the time-consuming alignment process again. Now, while I gave an example in which mitochondrial DNA was considered noise, this is just one example of many. Different experiments will introduce different sources of noise, and different protocols will produce different experimental artifacts. There is no one-size-fits-all when it comes to filtering. However, removing experimental artifacts or noise is a relatively straightforward idea in and of itself. A trickier filtration issue to deal with is that of ambiguous alignments. An ambiguous alignment is when one read aligns to multiple locations in the reference genome. The 12th column of the SAM file may contain an XS tag, which indicates the score of the best alignment score other than that of the current entry. XS:(I:10) would indicate that there is an alternative alignment with an alignment score of 10. Keep in mind that this only applies to files generated by Bowtie 2. Other programs have been reported to use the XS tag differently, so take the information in this video with a grain of salt. In some experiments, reads which align to multiple locations may be tolerated if the score for the secondary alignment is far weaker than the score for the primary alignment. In other experiments, all reads with any degree of suspicion are removed. The appropriate course of action would depend on the experiment from which the data had originated. There are many other factors to account for when deciding on the filtering steps for the read alignments, but these are some of the more general ones. And that concludes everything I have to say about the core pipeline.